Hey everyone, we would like to start the next session, and Dr. Ji-Young Park will have a session regarding the development of radiopharmaceuticals. So please welcome.
Hello, my name is Ji-Young Park in chemistry team in our nuclear medicine department. Today I will talk about the basic concept of the radiopharmaceuticals and development aspect of the Theranostics. So the clinical field and other cutting-edge radiopharmaceuticals and other QC program is already discussed for the previous lecture. So in this time, I will talk about the basic concept of the Theranostics or Diagnostics, and then the what can I say, that the traditional pharmaceutical for the using the diagnostics or therapy, and then we talk about the factor, about what factor is concerned about to use for the Theranostics or diagnostics experiment. So first of all, we hear the word about diagnostic and diagnostic. That is a combination word from the therapy and diagnostics. So like this concept is already studied and well researched some other fields, especially some other kinds of scientific field. So we normally hear that some kinds of concept from the other basic science. So in our field, we use some radioisotope-based diagnostic concept. For example, and technetium and iodine is some capitalizes example. The other things to use radioisotope-based diagnostic is the bioimaging guided radiotherapy. It means that some combination of some kinds of fluorescence dye or other kinds of imaging modality was conjugated to the radio-labeled one, so we can use that kinds of technique to detect some tumor or other kinds of pathological region. And the other part of the application is the optical imaging and laser ablation and surgery portion. It was so-called optodiagnostics. And nowadays, some kinds of engineering part was emerging, and then it was combination to our department, and then it was well studied about the nanomedicine. So it's a natural property of the nanoparticle, so it is really easy to access to other modalities. So combination to our diagnostic or other possible therapy radio stuff is more advanced research is possible in field of nanomedicine. So previously we studied about the radioactive compounds used for diagnosis and therapeutic treatment of human disease. So we say that our radionuclide was conjugated to the pharmaceutical compounds, and then we can easily access to the diagnostics or therapy. So more impressive property is that it was really safe and non-toxic for the human administration. So the traditional nuclear medicinal imaging agent was normally used as a metabolic imaging agent, and for example, FDG and iodine and phosphonated conjugate labeled technetium or rhenium is for example. So why I told you about this nuclear medicinal traditional imaging agent is that we use this kind of property to the more advanced diagnostic field because of we just use like this kinds of property, and then we just adding some kinds of diagnostic and therapeutic radioisotope, and then we can use type of this chemical to other advanced application. So right before we talk about some kinds of nuclear medicinal drug, and then we check the kinds of the the diagnostic some diagnostic agent. First of all, only RI administration is used for the diagnostic application. For example, we can easily access iodine or some lutetium that was possible to use some imaging and therapeutic efficacy. And then after that, we use some kinds of chelator for the more stable form in our administration to the in vivo system. So it was possible to use the therapeutic radioisotope. And then the next generation is we just use some drug and the radioisotope conjugated one. And then we can find some specific ligand and use some linker and use some chelator and easily label some radioisotope, and it was used for the diagnostic agent. For example, Dr. So already told you about the dotatate, dotato, and other kinds of prostate cancer imaging agent was some example of this. Nowadays, the cutting edge, the application of this diagnostics is the nano material. Some nanomaterial have its natural property, so we can easily add some additional modality to the radio diagnostics application. And then nowadays, some drug was also loaded to this kinds of platform, and it is possible to use some diagnostic efficacy. I think that all of you is already know about the glucose metabolism pathway, and we can use some FDG, and we use some diagnostic imaging about tumor. Because of the sugar is main only source of for the cell, and especially cancer cell gives higher sugar consumption compared to the normal cell, so we can easily access to the diagnostic imaging use this type of radiopharmaceuticals. And this is chart to mechanism of uptake to the cell using glucose. The natural glucose was uptake to using glucose transporter, so-called GLUT, and then it was uptake to the cell, and then it was go through the metabolic pathway using some kinds of enzyme. So after that, it was final product. It was shown some final product, carbon dioxide, and it was diffused from the cell to the plasma phase. But in case of nuclear medicinal drug, FDG, is also use some glucose transporter, so-called GLUT, and it was modification of carbon 2 site, so it is not go through the metabolic pathway using enzyme, so it was well uptake to the specific cells. This is normal image of human, and then this is patient image. You can see the clear spot in here, some kinds of some cancer cell was appeared in diagnostic image. Technetium is easily accessed and easily usable for our nuclear medicine field for diagnostic using SPECT imaging. Because of its half-life is adequate and its emitted gamma ray, and it was easily produced by generator, so we can use this type of radionuclide for our diagnostic imaging agent. Because of uranium, is also usable for same chelator using technetium because of its coordination number is relatively same, but its physical property is totally different because rhenium is possible radioisotope for the therapy because of its emitted beta particle. And more interesting things is it was also emitting some gamma ray, so it is possible to use imaging and diagnostic and therapeutic agent for using same kinds of chemical of technetium. This kind of therapeutic radioisotope is also from the generated system, but it is so because of is easily accessible, so we can use this kind of radionuclide for our therapy. For using this technetium and rhenium type of radionuclide, we preparing some cold vial, so cold reagent kit. In here, some ligand, it means that some targeting moiety or other kinds of chelator was already contained in here. And then for the labeling, some kinds of agent was also contained. And for the more stable labeling, so some stabilizer was also contained in this kit. After we preparing some kit formation, and then we can labeling like this kinds of process. First of all, we can preparing some kit, which was already sterilization, and then using some proper shielding system, we can easily owning some technetium or rhenium isotope from the generative system and then storing in this kit container. After that, we check the TLC. We already run about some importance about TLC check from the Professor Lee's lecture. In this step, we must check the TLC for the successful labeling or other kinds of free form, other kinds of different form we didn't expect. One is already checked right before the human injection. This is our facility to labeling some technetium systems. This is the lab space, and this is some generation, and this is our hood, and some shaker for the labeling, and then this is a heating bath. And we check some QC using some clean bench. We already see the more specific figure from Professor Lee's lecture, and this is a THC scanner we use, and this is some injection space. More specific structure of technetium pertechnetate is like this. So its radius and its physical properties are relatively similar with iodine. So it was well uptake to the thyroid, some kinds of gland. So the capitalized application of this technetium labeled one is the bone imaging. Because of phosphonate group was easily uptake to the bone, so and its chemical group was easy to access to labeling of technetium. So we used some kinds of phosphonate group for the labeling of technetium and imaging of bone. So using the methylene diphosphate, and after that technetium labeling, we can take some SPECT imaging, some bone imaging. And other application of these kinds of technetium radioisotope is the kidney imaging agent. Compared to the bone imaging agent, it was strengthened on lots of hydrophilic site in the edge of the chemical. So it can be possible to go directly through the kidney excretion. So this image shows some rapid kidney excretion using 99M technetium DTPA. So I already told you some technetium and rhenium is relatively similar physical property. So it was also showing some similar labeling process. Because of that kinds of property, we used the rhenium 188 to the DTPA labeling. But the DTPA labeling step of uranium is somewhat different from especially about the reaction condition. Technetium is really easy to labeling using DTPA just to mix about the DTPA kit and technetium solution. But in case of uranium, we must heating using water bath for 2-hour photo labeling. So diphosphonic acid derivatives, I already told you some bone imaging agent. So it can be possible to labeling with technetium 99M, and it is also possible to labeling uranium. So in case of that, it was showing some relatively same image, but uranium is possible to using some therapeutic efficacy. So other kinds of application of technetium and uranium technique is some macroaggregated albumin and sulfur colloid and thin colloidal and antimony sulfur colloid. It was so-called colloidal radiopharmaceutical. Because of its size and its stability of the intrinsic labeling of technetium and uranium labeling, it was used for the lung or liver or spleen or lymph node targeting. This is an example of technetium labeled sulfur colloid image. It was showing stable property using this kinds of labeling technique, intrinsic one. And this is technetium labeled antimony sulfur colloid to detection of the sentinel lymph node. And then I already show you some biphosphonic acid to the bone image, and then the relatively it was showing some relatively same image using the uranium and technetium, because of uranium also emitting some gamma ray, so it is possible to take some SPECT image, but the imaging quality is not good because of it was also emitting some beta particle. This is some clinical example about using some MDP and HEDP using some technetium and uranium. The other application of uranium is the lipiodor system. You can see the long hydrophobic chain. It means that it was well uptaken to the liver because of the liver naturally uptake to the hydrophobic molecules, and then it goes through the conjugated to the hydroxy group conjugation, and then it was make some more hydrophilic, and then it was excreted to the plasma. Using lipiodor, and we can make some uranium labeled lipiodor system, and then check the liver image for the using some animal. And this kinds of treatment was also already used for multi-center trial for liver cancer supported by IAEA. So out of 76 patients, in that 69 is stable disease, and 4 is complete or partial response, and 3% is showing some progressive disease. This is other example using labeling of radioisotope. So I already told you some drug labeled one. So we labeled it using F18 to the paclitaxel. So you can see the uptake to the target region. The next example is gallium-68. Because of gallium-68 is a metal radioisotope for the using some PET imaging system, because of it was emitting some positron and its half-life is 68 minutes, and it was really easy accessible to using generation system. So normally we use the gallium as a chelator using Ga-NOTA. So this chart is showing some possibility to use gallium as Ga-NOTA is a good labeling chelation of gallium, and then the other kinds of metal is possible to using Ga-NOTA as a chelating agent. Just a simple example about this figure. After labeling gallium using Ga-NOTA and then we adding some different kinds of metal solution, adding some different concentration. After adding 0.760 millimole of copper was added to gallium-NOTA labeled 1, the gallium was detached from about 13%. It means that its copper is possible.
Not detach, just inhibition.
Inhibition. It inhibited 31%. And then indium was inhibited to gallium labeling 67%, and iron was showing some 87%. It means that copper and indium and iron is possible to use NOTA as a chelator. So some example you already see from previous lecture using gallium system is gallium dotatate and other kinds of gallium PSMA agent. So using gallium labeling method, many kinds of peptide and protein and nanoparticle is possible to labeling. And also small molecule is possible to labeling use same kinds of method. So we call that this kinds of method is the indirect bifunctional chelating agent method. So we call the BFCA. Some terminal about peptides or other kinds of small molecules and other kinds of ligand we can conjugate to this kind of chelator and then easily to labeling of radiometals. For example, the gallium and copper and lutetium and indium also. So some factor about BFCA is to make some stable complex. And then in the labeling condition is must concerning about the pH and temperature relation time. And we must consideration about stereochemistry of the radio labeled one. So some kinds of radiopharmaceutical already we see some previous lecture. And this is another example about using NOTA RGD1. This is for the angiogenesis imaging using the RGD ligands, and then it was conjugated to the chelator in here, and then it was chelating to gallium radioisotope. So left figure showing some without blocking, it means that we just injection to the labeled one. So you can see the red arrow, some tumor region, it was take well, but we co-injected it to the blocking agent using RGD. It was not taken to the tumor site. This is the final subject in my presentation. Some considerations for the radionuclide therapy. Because of radionuclide therapy is used for therapy using radionuclide, we must choose adequate radioisotope. Previously, Professor Lee and Seo already discussed about the alpha particle and beta particle and possible tumor to use that kinds of radioisotope for we can consideration about the tumor size and what pathology we target and we want to therapy. And it was totally different from external radiation therapy. So we must check the dosimetry for our more safety concern. And we must concerning about the with or without carrier because of some aspect of the dosimetry. We check this kinds of consideration, and then we choose the targeting, and we must consideration about effectiveness of this kinds of radionuclide therapy, and then choose the proper carrier. It means the chelator and bioactive molecules. After that, we check the dosimetry, and finally, we must consider the cost effect. This this table showing some radionuclide already used in the therapeutic application, and iodine 131 was emitting some beta and gamma, so it is possible to take some SPECT image, and it is possible to therapy. And yttrium showing only beta emission, so it was possible to only therapy. Lutetium also emitting some gamma ray and beta particle, so it is also possible radioisotope for diagnostic and therapy system. Uranium is also same. So I already show you some importance about the chelation, because we can easily access some same structure for the diagnostic system and therapeutic system. In some case of NOTA, it was possible to labeling about 68 gallium and 64 copper and 177 rotation. It means that we can use these kinds of chelator, and we use this kind of chelator for our diagnostic application or therapeutic application that was already have some same structure. So, and DOTA is also possible to creating yttrium and lutetium and actinium and copper. So it means that we use some copper, and if we use some actinium or lutetium or yttrium for the choose some adequate radionuclide for each kinds of application, we can easily possible to take some diagnostic images and have some check the efficacy of the therapeutic aspects of radionuclide. So we must know about the structure, and we must know about the possible radioisotope labeling of each kind of chelator. This is end of my presentation. So if anyone have some question, fully writing some chat box. Thank you.
거기 밑에 보면은
이거 말씀하시죠.
네, 거기 아니 그 스카이프고 저쪽을 가봐. 저쪽 화면. 거기 있다. 거기에 참가자 봐봐. 그거 아니야. 닫아. 스카이프를 닫으라고. 이쪽으로 올라가봐. 참가자에 거기 근데 채팅 위에 있었는데 사라졌어. 위로 올려봐. 더보기. 저쪽 맨 끝에 더보기. 채팅. 그다음에 이거 뭐라고 했어? Is there any question?
Yeah.
Okay, then we can have a break, 10 minutes break, and then I think in 2:40 we will start 2:40. Is that right? In Korean time, 4:40. Okay. Okay.
Okay. Thank you, Professor.
Thank you very much.
Hello, Dr. Park Ji-young.
Yes.
Hello, Dr. Park Ji-young. This is Jushimuddin. How are you? Thank you for your nice lecture.
자신.
자신.
Yeah.
Thank you.
Thank you.
Yes.